Madam Pro-Rector.
Yep. Hereby, I open this academic ceremony in which Gennaro Martucci will defend the academic thesis, Challenges in Extracorporeal Membrane Oxygenation, Precision Management to Improve Survival. I'd like to welcome everybody, of course, the candidate, Mr. Martucci, the members of the supervising team, Professor Lorusso and Dr. Raffa. Unfortunately, Professor Maase cannot join, his apologies, but he's busy with some problems in the operating room. I also like to welcome the people who are watching this live stream. I also like to welcome the members of the degree committee, who I will introduce later on. Mr. Martucci, may I ask you to present a summary of your study and the conclusions of your thesis?
Dear Pro-Rector, dear members of the Corona, dear family, friends, and audience, in the coming 15 minutes, I will shortly summarize my thesis project. First, extracorporeal membrane oxygenation, also named ECMO, is a life support system. It is able to replace the function of the heart and/or of the lungs. It is a complex system composed by cannulas, tubing, a pump that is able to replace the heart function, an oxygenator where the gas exchanges occur. And all this is an modified extracorporeal circulation. So the blood is exposed to an external surface and is at risk of coagulation. That's why we need an anticoagulant to avoid thrombosis into the circulation. ECMO has two main configurations, the veno-venous configuration and the veno-arterial configuration. And these different types of ECMO correspond also to their main indication. In case of veno-venous ECMO, the blood is drained from a big vein, usually from the abdomen, and then is returned to the patient via another big vein. So this is an in-series circuit on the venous side. While for the VA ECMO, the blood is drained always from a vein, but then returned to the patient via an artery. This can be a central or peripheral artery according to the type. But anyway, the VV ECMO is more indicated for the acute respiratory failure. So VV ECMO is for respiratory support, and the leading cause is the acute respiratory distress syndrome that recently has been better defined by the Berlin definition as a syndrome. But still, there are many causes for acute respiratory distress syndrome, from bacterial pneumonia to viral pneumonia, and in the last year, COVID-19 has challenged this topic. While the veno-arterial ECMO is mainly indicated for cardiogenic shock, so a low flow state from the cardiac side. But anyway, the veno-arterial ECMO is also able to give some respiratory support. So this is a complete cardiorespiratory type of support. Moreover, we know that in the last year, in the intensive care unit, the number of patients admitted is absolutely increasing. And also the request from the public opinion, from governments, family, for an improvement, a reduction in mortality. And here in this table of data from the Netherlands, we see that probably the aging of the population is accompanied by an increase in the hospital admission. And this also poses some question of sustainability for the critical care. But on the opposite side, we know that ECMO is able to reduce short-term and mid-term mortality. That's why probably the number of ECMO runs and the number of centers, as we see from the data from the Extracorporeal Life Support Organization, is absolutely an exponentially increase in the last year. And looking at the outcomes, here we see the in-hospital survival. Probably the results are already very good, but there is some room for improvement. Not saying about the COVID-19, that in the last 2 years was an unprecedented occasion for ECMO use, as the data from the ECMO COVID study led by the Maastricht Medical University is demonstrating. Now we are at almost 6,000 cases just in Europe and just the registered one. And the in-hospital survival is at about 50%, with some variability between the different COVID waves over the months. But so how we can improve the survival? We elaborated the concept of ECMO precision management. That means putting together all the knowledge to increase survival or reduce mortality. And first, the topic, the debated topic is probably the coagulation and the anticoagulation balance. Then the daily blood flow management, since we know that the blood flow during ECMO is the real support that we can give to the patient, and it is still undefined how much support we have to give in the most severe phase of the disease and when the weaning is ongoing. Then there is a technology improvement. So the community should be ready to profit from the rapid improvement and rapid changes in the biomaterials and try to apply them in a fair way. Then the topic of hemoglobin and transfusion is relevant since this is an extracorporeal circulation, and hemoglobin is an important carrier, obviously, for oxygen. Then the complications that are one of the main pitfalls during ECMO, since complications are frequent and are an important cause for morbidity and mortality. And finally, last but not least, the proper indication. So we have to find a way in the next years to recognize the patient that has a real chance to improve survival thanks to ECMO. And these are some of the examples from the manuscript in the thesis. The first one is about anticoagulation and transfusions during ECMO for respiratory failure. This was a cohort study on 82 patients. And in this study, despite we introduced a very low heparin protocol, we anyway experienced a consistent number of episodes of bleeding, 34, but this was a major bleeding, so needing a big amount of transfusions or an active action in just 14% of cases. And this was also the occasion for a publication of the protocol of anticoagulation at ISMET based on low heparin dose, frequent APTT coagulation monitoring, and daily antithrombin monitoring. We also analyzed the transfusions during ECMO, and we recognized that the acute kidney injury, the use of vasopressor, as well the use of an older device with less monitoring was associated to an increase in transfusions. And this was important because transfusions, together with the occurrence of the septic shock during ECMO and the hospital stay pre-ECMO, were major determinant of mortality, and this was also confirmed by another statistical analysis. Following the same path, we also performed a survey on transfusions in ECMO. This was an international survey endorsed by the European Society of Intensive Care Medicine. And the main achievement from this survey was that in ECMO, the patients were kept with a hemoglobin higher than in other critically ill patients. And interestingly, in the centers with a higher caseload per year, in this case, more than 24 cases per year, hemoglobin was at a lower level and a transfusion structured protocol was adopted more frequently. But even though in this busy image, we also asked for other parameters that the respondents would consider before transfusions more than hemoglobin. And despite a huge variability, the majority of respondents were in agreement that more than hemoglobin, we should also think about other parameters like the blood flow, the fluid balance, the global perfusion before administering transfusions. Then we also considered some of the main complications, and these were the gastrointestinal complication. I said is a neglected topic, probably because when we care for the cardiorespiratory side, maybe we forget about other side. But in this case, we had 14 patients on 139 total VV ECMO patients with gastrointestinal bleeding and treated with advanced endoscopy. As always in ECMO, even an unimportant bleeding may become a major bleeding. And in this case, in 50% of cases, the bleeding was major. And despite the similar pre-ECMO characteristics, with the action through the advanced endoscopy, there was no difference in mortality. The ICU discharge and the 6 months mortality was similar between patients with gastrointestinal bleeding and without. And this was even compared to the data from the International ELSO registry in the subset of patients with gastrointestinal bleeding. Then we also considered the intra-abdominal hypertension. This is a frequent complication in critically ill patients with also a complex medical management. The most important thing is when the intra-abdominal pressure stay high for a long time, it can worsen to the abdominal compartment syndrome, and syndrome characterized by organ dysfunction associated with a sustained pressure above 20 millimeters of mercury. In the case of ECMO, this is really important because we say that ECMO drains the blood mainly from the abdomen. So maybe there can be an ECMO blood flow impairment. And when the abdominal compartment syndrome is prolonged, a decompressive laparotomy is indicated, but this carries a lot of problems as bleeding and infection. That's why we proposed the use of a non-invasive treatment, an intraluminal decompression via the total water-assisted colonoscopy that probably could apply in this type of patients. And finally, we also focused on the characteristic of patients with ARDS that, as I said, is an acute inflammatory disease with an injury and then an overlapping phase through the chronic lung repair. This is a syndrome that represents more than 10% of the ICU admission. So a better understanding of characteristic of the patient is probably mandatory. And in this case, microRNA, they are some small non-coding RNA able to modulate specific pathways. And they are changing in case of the disease. And they are part of the communication between cells in case of inflammation. All saying that these are probably good biomarkers for inflammatory disease. We started from a panel of 754 microRNAs, and with different techniques, all the changing microRNAs were able to divide the patient into two groups. And finally, we see that the two groups were with different severity. And the 11 microRNA that were altered in our pilot cohort were involved in the tissue remodeling process, in the regulation of the immune system, and in the regulation of the coagulation. So all topics that are plausible and important pathways for the ARDS. And finally, in conclusion, we can say that hemoglobin is fundamental during ECMO, but is just one part of the circulating blood volume. About anticoagulation, low heparin infusion probably is feasible, and antithrombin has a high rationale during ECMO, but still it should be tested in large prospective studies. To treat complication in non-invasive way is a topic to be explored in the next years during ECMO, and the circulating microRNA are a potential biomarkers for characterization of ARDS patients. So precision management in ECMO means probably just the integration of multidisciplinary care that hopefully could conduct to better outcomes. There are some future directions, since during the years of this PhD, an international multi-center prospective study was conducted, and I hope this will give to the community some results in very soon. And then 2 new studies on microRNA are on the way. And for sure, the direction is the care of the critically ill patients according to the best and updated evidence. So with that, I want to thank you for your attention, and I give the word back to the Pro-Rector.
Thank you very much. I would like now to start with the opposition. And the first opponent is Professor Wesseling from Maastricht University Medical Center. Professor Wesseling was chair of the assessment committee. Professor Wesseling.
Thank you very much, Madam Pro-Rector. Dear candidate, first of all, I would like to congratulate you and your supervisors for a very interesting thesis and a very interesting body of research on ECMO. It was a real pleasure reading your thesis, and especially as a pulmonologist, I have a number of questions related to chapter 2. In this chapter, you've studied the implementation of a protocol for anticoagulation and transfusions in patients who are being treated for ARDS with ECMO. And your main outcome measure is survival in the ICU and at 6 months after the ICU stay. As a pulmonologist, obviously, I'd be interested in having a little bit of an idea how such a protocol influences the outcomes in terms of lung function or quality of life. That has not been the subject of your study, but can you elaborate or can you speculate a little bit on how manipulating transfusion or anticoagulation protocols could influence outcomes in terms of, for instance, lung stiffness or lung function or quality of life?
Highly esteemed opponent, thank you for this question. So the main outcome that we considered was mortality. For sure, one consideration that we can do about the lung function and, let's say, the quality of life is the impact of our protocol for anticoagulation and transfusion on the stay on ECMO and on the ICU stay. So in this case, I believe that to reduce transfusions may probably reduce the ECMO stay, or at least the aim is to reduce the ECMO stays and render the ECMO weaning earlier. So in this way, we should reduce the time for inflammation and probably try to follow a more rapid recovery for the lung function. I can just add that this 6 months survival was assessed in patients that were all already at home. So the one that were alive at 6 months were all out from the hospital.
Would the outcomes be the same if you had done this study in COVID because obviously your research was done before the pandemic. Could you think of how the outcomes might be with COVID pneumonia or ARDS caused by COVID?
Yes, I can imagine that the outcome would have been a lot worse, I think. And the reason is first because of the data from our study, and because as you remember, I said the hospital stay was an important determinant for mortality, and at least in our cohort of ECMO in COVID, but I think that's a process ongoing worldwide. The hospital stay, even the hospital stay for every COVID patient before ECMO is quite long. So we had also recent data saying that the length of mechanical ventilation probably does not impact the outcome during COVID, but the hospital stay before the cannulation for ECMO is probably paramount to determine the outcome. And just as you said, from the pulmonary function, probably 2 or 3 weeks with high respiratory effort and high work of breathing, even non-invasively, even without intubation, may be worse and may impact a proliferative state in the lungs, and consequently also a fibrosis process that in many times, and probably in higher age, became irreversible after 3-4 weeks.
Thank you very much for these answers. I give the word back to the Prorector.
Thank you, Professor Wesseling. The opposition will now be continued by Professor Brody from Columbia University, New York. Professor Brody was also a member of the assessment committee. Professor Brody.
Thank you, Madam Prorector. Dear candidate, first of all, congratulations, because you've done what is really important work, which already has influenced the field substantially. And as you alluded to at the end of your talk, the PROTECMO study will have what I think is a major impact. So my questions for you are really around what comes next. What are the next important studies? And the first is with your very nice data on the factors associated with transfusion requirements in veno-venous ECMO for ARDS, along with the survey on transfusion practices, and of course, PROTECMO. What is the next study that should be done? Is it a randomized controlled trial? How do we determine the optimal transfusion strategy? And should it be a hemoglobin trigger like in the TRIC trial from 1999?
Hi, esteemed opponent, thank you for this question. Really, the take-home message for me after these years involved in the PhD and also looking at some worldwide data, the only conclusion is that probably the hemoglobin trigger is not worth to explore as a single trigger. So the only way that we as a community can manage the topic of transfusions of hemoglobin, that means also not only harm for the patient because of transfusions in the immunomodulation, but this means also something about the costs of transfusions and also the social costs for a limited resource like the blood products, and that we should take together many aspects at the same time, and starting from the standardization of practice and also the standardization of an ECMO center as a whole, as a different as a practice, we should consider the hemoglobin just one part of the circulating blood. So we cannot consider it without a project on the amount of blood flow that we want daily achieve, and the type of blood flow that we want to achieve when the support needed is full or when we are in the weaning process. So the main message that I've received is that we have to keep together hemoglobin, blood flow, and probably as a main factor, the fluid balance to keep the patient the more stable and the blood flow the more stable, to give the patient time to recover, and also to the other treatment that we can put in place to be able to act and let the patient recover.
Thank you, Mr. Candidate. I like that answer. My second question is your work delineating the current use of antithrombin 3 and veno-venous ECMO in your review demonstrates no real consistent use of antithrombin 3 across centers, which was important as a finding, but not so surprising for those of us in the field. So should we plan a prospective trial of the use of antithrombin 3 and what might that look like as well?
That's an important question. Actually, we use antithrombin, and we have to say that worldwide, the use of antithrombin is about 9–10% of the ECMO centers. And historically, probably this is more used in the pediatric population. So centers doing also pediatrics probably are more prone to use antithrombin also in adults. In my view, I think we should do that, but the main reason probably was that antithrombin was in fashion 20–25 years ago for the anti-inflammatory properties, and then some trials unfortunately failed to demonstrate its usefulness in case of sepsis and septic shock. But the rationale for use of antithrombin is really evident for the glycocalyx protection, for the endothelial protection, and also because heparin is our main anticoagulant, and without antithrombin it cannot be useful. So to your question, I answer yes. In my opinion, we should go for a randomized trial on antithrombin, but probably we still need some preliminary data to understand in which setting of population we should apply that. Also considering that now we have also other different anticoagulants that like direct thrombin inhibitors and others that are on the market. So probably we should not consider heparin as the only option.
Thank you, Mr. Candidate. I give the word back to the Pro-Rector.
Thank you, Professor Brody. The opposition will be continued by Professor Shanongowski from the Medical University in Vienna, Austria. And Professor Shanongowski was also a member of the assessment committee. Floor is yours.
Thank you, Madam Pro-Rector. Mr. Candidate, I congratulate you for your concise presentation, for the body of literature that you have produced over the last years, and for your efforts and prospects that you have produced with your intense actions towards the PROTECMO data that are currently being investigated. My first question is concerning the microRNA data that you have looked into. Now, given the background that you obtained, when we look at the COVID population, which certainly came up after your studies, do you believe that microRNAs would be suitable to answer the questions, are there really subphenotypes of ARDS in COVID patients?
Esteemed opponent, thank you for this question. And for sure, COVID-19, as I said, is challenging the classical ARDS definition. Actually, it is an ARDS from the clinical side. All the numbers are in the right place for the ARDS definition. But let's say the pathophysiological image of ARDS due to COVID is probably different, at least at the beginning of the disease. I'm not speaking about the COVID-19 when there is subbacterial superinfection, as we see quite often in Sicily. So looking at the microRNA and looking at the subphenotypes, probably in the COVID-19, we still have not explored enough the subphenotypes, since we see that many patients, or at least in a certain phase of the disease, many patients have a high inflammatory state, but some treatment that are able to reduce the anti-inflammatory state, like the hemoperfusion, has failed to impact probably the mortality. So in this picture, the subphenotypes can probably divide or differentiate patients with COVID in different subgroups, and microRNA potentially may be a part of this puzzle. But these are all preliminary data, and the main limitation for this data is that to work with microRNA is currently very costly for every institution. So probably we are still in a germinative phase, so we cannot profit from big numbers in this case.
Thank you for the answer. Madam Prorector, is there still time for another question? I take that as a yes. Mr. Candidate, it's in the title of your thesis, Precision Management, and we are talking about transfusion. I would like to ask you about the transfusion of thrombocytes. There's in my opinion, quite arbitrary threshold for thrombocyte transfusion in international recommendations. If you were to individualize and personalized transfusion threshold, which would be the variables that you consider when evaluating at which threshold this given patient should be transfused?
I would consider the number of thrombocytes, the trend of the thrombocyte over time, and also the characteristics of the oxygenator and pump. Since if we transfuse thrombocyte to an ECMO patient, we take a high risk of circuit thrombosis and reduction in the blood flow and reduction in the support that we offer to the patient. So I understand why there is an arbitrary threshold, because during ECMO we can experience very low platelet count without transfusion, without bleeding, and also almost normal platelet count with transfusion. So in this case, my answer would be that first we should know more about coagulation. So maybe we should monitor the coagulation with viscoelastic tests to understand the impact of fibrinogen and also the coagulation factors, and then try to give transfusion according to when we want to change our circuits and our oxygenator.
Thank you, Mr. Candidate, and I give back to Madam Prorector.
Thank you very much, Professor Shalonkovsky. Opposition will be now continued by Professor ten Kate from Maastricht University Medical Center, and Professor ten Kate was also a member of the assessment committee. Professor ten Kate.
Thank you very much, dear candidate. I also read your thesis with pleasure, and I want to congratulate you with this also colorful but very insightful work. And the previous questioning was already quite challenging, and maybe I can continue a little bit on the path that was started by Professor Brody, where he discussed anticoagulation and the role of antithrombin concentrate. I learned from your answers that in Palermo, antithrombin is currently used in ECMO situations. Could you, as a first point, tell me which indications you use for the use of antithrombin? Is that mostly in the pediatric population? Do you use a certain threshold, or how do you use it?
Esteemed opponent, thank you for this question. Actually, in our protocol, we measure antithrombin daily. So we have to remember that antithrombin is not directly measured, but this is a percentage of the level of activity. And we use it, we are an ECMO center mainly for adults, but also for all the southern Italy. We serve all the southern Italy for pediatric ECMO, but the number of cases is luckily very, very, very few cases per year, 1 or 2 cases in pediatric population. And so we use it in the pediatric population and also in the adult. And our limit is 8%. From the books, we know that the normal level is between 80% and 120%. And the reason why we give it is just the rationale that the heparin works thanks to the antithrombin. And the more heparin we give, the more we think we could experience bleeding, also an unseen bleeding from the gastrointestinal side, from the cannulation side. While in our idea, the administration of antithrombin can be a more physiological way to balance anticoagulation and coagulation.
That's also a point that you discuss in chapter 7, and that's quite intriguing, but because it suggests, at least that's what you think, is that by giving additional antithrombin, you can somewhat downplay the amount of heparin that you give, and that way perhaps provide a better hemostasis or less bleeding risk in your patients. Do you think that there is good evidence for such an assumption, that by changing the ratio of antithrombin to heparin, you can adjust the bleeding risk?
Probably the classical evidence is not yet available for us and for the community. But in my view, or at least in the view of our group, this can be a chance to reduce the impact of bleeding complication during the ECMO stay. All this was also challenged anyway, and in our experience, this was working quite well with low heparin dose, no frequent need for interruption of heparin, while all this was challenging then in COVID-19 patients, where this type of anticoagulation didn't work in the effective prevention of major bleeding. And so, as we said before, probably COVID-19 are really a special population that we cannot consider always together with the standard, with the patient on ECMO that we considered standard till two years ago.
I agree that the COVID setting, of course, was a very exceptional situation. Did you also try higher amounts of antithrombin transfusion in COVID patients, or to see whether you could provide additional cellular protection with supra-therapeutic doses, or didn't you try that? It's just out of curiosity.
No, we actually did not. But the reason was quite simple. We were flooded by the second wave of COVID, And we arrived to 19 ECMO at the same time, and we are a center doing usually 2-3 ECMO per day. And for sure, we ran out with several drugs, and sometimes also with antithrombin. So it was really unfeasible to get to supratherapeutic doses in this case, because it was not available in the fridge.
I fully understand. Maybe a very short question, if I'm allowed. The last question, Madam Pro-Rector, is that okay?
No, there is no time.
No time left.
Oh,
okay,
I'm sorry. I'm sorry.
Plenty of questions left. It's an exciting discussion, but I will give the word back to you. Thank you.
Okay, I'm sorry, but because of the time, we have to continue. And the next opponent is Professor Pappalardo from Santi Antonio e Biagio Hospital in Alessandria, Italy. Professor Pappalardo.
Thank you, dear candidate. Let me first congratulate for the excellent work that you have provided, and which shows a great amount of work over the last years in the field of ECMO. I would like to ask you about your very intriguing perspective of personalized medicine and precision medicine. In this regard, can you envision such a strategy also in terms of the ECMO system technology itself? So to change the perspective from the patient to the technology?
Highly esteemed opponent, thank you for this question. Actually, I agree this is a possibility to switch from a single type of technology used in every patient and one size fit for all to a personalized approach. Even though now we know that this separation probably is excessive, the separation between the ECMO ward as a respiratory support and the ECLS as a cardiac support is probably not really rational. And there are many stakeholders, some of them are in the corona, saying that probably one of the problems of ECMO during COVID was that the cardiac support was adopted in too few cases. And we know already that many times we have also a cardiac impairment in case of respiratory support. So that's why some hybrid configuration were born. But mainly, the most important thing is that in my view, is the multidisciplinary approach. So even though during respiratory support, we should consider adequately the cardiac function and maybe sustain the right ventricular dysfunction whenever it appears and whenever is in place, because without cardiac recovery, probably we will not be able to wean off the patient anyway.
And just to further speculate on this topic, I think that this cardiac involvement in lung failure might also be a matter of delayed implantation. So how much do you think of the adverse events we face in daily practice with these ECMO patients might be at least mitigated by an earlier adoption of ECMO?
Yes, I agree with you. Probably there are many data coming up from the last years that an early use of ECMO is probably better, and switch from a rescue therapy to a therapy able to support the patient even before the intubation or even before an excessive critical state. There are recent data from Germany that they are using ECMO, respiratory ECMO, instead of intubation, with some positive insights. And as said before, probably to avoid a prolongation of dyspnea and subjective respiratory effort and work of breathing may be positive for the outcomes and may reduce the intrapulmonary stress and strain, and so reduce the inflammation and the following fibroproliferative processes.
Thank you. Madam Prorector, I give you the word back to you.
Thank you very much. The opposition will be continued by Dr. Arkady Pane from Palermo, Italy.
Thanks, Madam Prorector, giving me the words. Mr. Candidate, first of all, congratulations for the excellent work done for your thesis and for the body of work you did that you will do probably in the future in the field of ECMO. The question is related to the chapter 3, and I want to ask you how you can explain that cannula design impact on transfusion or outcome.
Well, esteemed opponent, thank you for this question. Actually, in this Maastricht, the cannula design was studied in the light of transfusion. Actually, the proposal was for a tip-to-tip coated circuit, and with a coating that is reducing the, let's say, the coagulation and the thrombosis. One of the pitfalls of the study, and it was also a limitation explained in the study, was that the group with bioline coating, in this case, had some larger cannulas, and this was significantly higher than the other group. So in this case, probably the larger cannulas were able to reduce the pressure in the circuit and can guarantee a stable circuit, a stable blood flow with lower pressures. And these are probably one of the reasons why we can reduce the hemolysis and also the shear stress on the blood from the cell point of view, but also the other coagulation factors. And so probably this is the main reason why the transfusion amount was lower. But for sure, also the use of cannulas with an anticoagulant coating probably can, in the early phase, at least in the early phase, reduce the use of heparin, and so reduce the need for anticoagulation.
Thanks, dear candidate. I'm fine with your answer, and I'm giving back the words to the prorector.
Thank you very much, Dr. Arkady Pane. The opposition will be continued by Professor van der Horst from Maastricht University Medical Center. Professor van der Horst.
Thank you, Prorector. Mr. Candidate, I want to congratulate you and your team on an inspiring manuscript, but above all, on the pictures for every chapter. In chapter 9, 13 patients were supported by VV ECMO for ARDS, and in these patients, blood was drawn to screen for the expression of over 700 circulating microRNAs using high throughput qPCR. And based on the study, you concluded, or your team concluded, that the blood levels of 13 were altered in patients with severe RDS. My first question might be simple. What was your definition of severe ARDS in this chapter?
Esteemed opponent, thank you for this question. Actually, the definition of severe ARDS was according to the Berlin definition. So we considered all the all the data for ARDS, and then the PF ratio, so the PaO to FiO2 ratio was under 100 in all these patients, and they were severe from the pulmonary point of view.
Okay, yeah, because I did understand that you divided the two groups, cluster A and B, that one was more severe than the other.
Yes, the difference was about, generally speaking, severity. So the 13 patients were self-dividing according to the microRNA distribution. And then with two different analyses, one in silico and one in vivo, the different microRNA were separating in a way completely similar the patient into two groups. And the two groups, when we went to compare the two groups, they were different for the SAPS2 at admission, and also other two severity score applied to the ECMO patient, so the PRESERVE score and the RESP score. And this was a separation not driven by randomization or clinical decision, but we went just through the platform for the 754 microRNA. And so this was quite interesting, because with two different analyses, the same non-coding genes were involved.
Okay, yeah, thank you, because I thought maybe you thought severity was differences in the SOFA score or in the SAP score. So that was my question. You also tell us that there were different underlying causes like viral and bacterial pneumonia and also severe polytrauma. How were these patients divided between the two clusters? Because in your table 1, they are not presented. So do you know that? If one group was more pneumonia and the other more trauma, yes or no?
No, they were randomly. Sincerely, they were in both groups. Actually, bacterial pneumonia and viral pneumonia were the more consistent numbers, and then there were one or two polytrauma. But I think we tried initially for the pulmonary and extrapulmonary division, but with such a little amount of numbers, then we considered trivial to make this differentiation. Probably we need more data to say something relevant on also on the etiology.
Yeah, because and the last, you present us the scores and the subs. So like SOFA and SAPS, and that's to do something with comorbidities, but like for renal failure, then it's equal between those groups, and like renal replacement therapy is equal. So were the other comorbidities divided differently? Because one cluster seems to be more sick, even more sick than the other.
I think the reason was about the inflammatory state and the hemodynamic impairment. You know that in SAPS2, probably the hemodynamic impairment has a high impact on the number. And when we consider the RESP score and the SAPS2, probably the other difference was about the number of days before ECMO, that in this case is probably quite relevant to the outcome. And I would not consider probably the age as something impacting this topic, since we have quite homogeneous age for our patients. They are usually between 40 and 60. We don't go too much high with the age, and luckily under 40 years old, it's quite rare, the use of ECMO.
Okay, because now I can build it up, because I was lacking this information. because you say, okay, cluster A and B are divided by hierarchical clustering, and then they look in table 1 the same, but because the underlying cause or maybe the hemodynamic state did differ a little bit, I would ask you, and that's more or less my final question, if you think this is purely driven by an underlying pathophysiological mechanism that we can't see, or in this group of only 13 patients, maybe still the differences at baseline are the main drivers for your outcome.
I would not agree with the second options, since we in this study we also considered the baseline data for the outcomes, and the baseline data were not so much impacting the outcomes. That's why we went back. That's why we went back to the separation of the microRNA. And so the interesting part is that the patients were divided automatically by the microRNA. The outcome were quite similar. Actually, in this patient, they were the majority were alive at the end. It was a fortunate period of ECMO for us, some fortunate months on ECMO, but the microRNA were involved in some biological pathways that are really plausible being important during the lung recovery.
Thank you. For the sake of time, I think for your next publication, if you present this data, I think then you can go to the real high-end journal, because you're onto something. And I see you're proceeding with this kind of research already. So I'm very happy with these answers. Prorector, it's up to the other questions.
Thank you very much, Professor van der Horst. We now go to the last opponent, which is Dr. Delnoy from Maastricht University Medical Center. Dr. Delnoy.
Thank you, dear candidate. I also read the thesis with enthusiasm, and I think the thesis does great credit to the fact that ECMO treatment has a lot of hooks and angles and is quite difficult to run sometimes. And one of the topics we haven't discussed yet are the topics of the gastrointestinal complications. In chapter 8, you describe a way of managing intra-abdominal hypertension. Now, I read the article with great interest, and it was not clear for me what the time interval is between the measurement of the intra-abdominal pressure and the performing of the colonoscopy, especially since in most patients multiple interventions are being performed. And I would like to zoom in on neuromuscular blockade, because that's a prerequisite for an adequate measurement of the intra-abdominal pressure. And the timing for me was unclear on the numbers when you perform the measurement and when you perform the colonoscopy.
Team opponent, thank you for this question that make me clarify something about this manuscript. So about the measurement, we try to stick to the Society for Abdominal Hypertension, Abdominal Compartment Syndrome. So we use the measurement in with 50 ml of physiologic solution. But this is performed many times per day. And as I said, the medical management is wide. So we try to afford the colonoscopy after adequate sedation is in place. The carrier is increased, so transfusion has been performed. We try to reduce the fluid balance through the continuous renal replacement therapy, and for sure, neuromuscular blockade are in place, as well as also gastric detention via gastric tube. Then, when the amount of vasopressor is really high, and we are in danger. We think that the patient was going to be in danger because the blood flow was not able to be anymore sustained by our infusions. We decided to switch to the colonoscopy, and this water assisted permit to do the colonoscopy without preparation, without moving the patient. So we did not need CT scan, we did not move to the operating room. And also from the surgical point of view, our team was scared about the real possibility of these patients to survive to even to the intervention. And so we did not prove, comparing to the decompressive laparotomy, the importance or the effectiveness of this strategy, but for sure it was able to reduce the abdominal hypertension without any complication and without bleeding. And in any case, then the stay in the ICU was continued for several days after this procedure. So these are just germinative data, but they were effective, and they can be effective from the rational point of view.
Okay, thank you. So the only difference between the measurements before and after is the colonoscopy performed in between the two measurements.
And the beadle has told us that it's hora est. So the time for your defense has passed. You can answer Dr. Delnoy's question later on, maybe. But the time for the official defense has passed. So the degree committee will now withdraw to discuss the quality of your thesis and your defense. And I request that you and your company await the results of our deliberations and our return. Gennaro Martucci, the degree committee here present online has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Lorusso is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor. Professor Lorusso?
Yeah, thank you, Madam Prorector. So, Dr. Martucci, do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
So by the authority vested in us by law and in conformity with the decision of the committee here present online, I hereby confer upon you, Gennaro Martucci, the degree of doctor and grant you all the rights attached by custom and law. As evidence of this, you will soon receive the degree certificate signed by the rector, the secretary, and the supervisor, affixed with the official seal of the university as shown by the beadle now. So, may I, Madame Prorector?
You can continue.
So, dear Dr. Martucci, dear Gennaro, it is really a pleasure for me being here with you. And first of all, let me congratulate you, your family, friends, as well as colleagues and the team in Palermo for the brilliant work done, including obviously the thesis realization, your presentation and defense today. Really impressive. I still remember the first time we met at an ECMO meeting, during which I immediately perceived your passion, motivation, and dedication to this challenging yet fantastic field of medicine, which is extracorporeal life support. I saw at that time a young clinician looking for opportunities and collaboration, eager of reaching an in-depth involvement in clinical, but I would like to say sometimes also in basic, research in the ECMO setting. You address your attention and efforts, and this I think I really like that from the very beginning, towards the patients affected by respiratory compromise treated with ECMO. Your continuous and dedicated work investigated peculiar aspect of such a treatment, always in the light of finding even the limitation. And I would say sometimes, as we discussed many times, also about the mistakes we do and of the current strategies, while having always very clear in front of you to explore and find better ways to take care of these challenging patients. The recent era, as we all know, unfortunately told us how much this kind of therapies and how extracorporeal life support could have been relevant, could be helpful and critical for patients with refractory respiratory compromise. Unfortunately, not of them, but certainly we could save many lives. The COVID-19 outbreak prompted us in an unprecedented difficult time, desperately looking for effective therapies in patients sometimes without apparent help or means to recover, and ultimately experiences what ECMO represents, sometimes really the last resort, and in many circumstances successful, fortunately, in desperate otherwise patients, once we would have had or considered irreversibly ill and without hope. This fortunately proved not to be true, with almost 50% of patients, based on the worldwide experience, saved by this technique. Again, underlining how much science and technology are so important and so much embedded and critical together for the today and tomorrow medicine, as well as treating otherwise hopeless patients. Your work, therefore, has an additional value since highlighted some of the current limitations of this therapy, shedding some new light towards certain aspects of the ECMO support in patients with unresponsive or refractory RDS and requiring such a therapy. You have a clear profile of a clinical scientist whose work and reputation has already been recognized by the international community. Having you as invited speakers, international congresses, or contributor to multi-center study, or even, as you showed us, already as a principal investigator of multi-center worldwide trial, for which I personally look with admiration and happiness since I witnessed your constant growth clinically and scientifically. I've been therefore really delighted to having accompanied you during this successful pathway of your scientific career, and that you chose the Maastricht University and our research team to be with you and to be supervised during such a special journey. I am sure that we will listen and read more and more in the future about your work and scientific successes and achievements. And I'm also sure that our collaboration will continue stronger and more fruitfully. Again, on behalf of the Maastricht research team and university, I congratulate you and wish you all the best for your future. Al Mayora, Gennaro. And I give the word back to Madame Prorector.
Thank you. Dear Dr. Martucci, also on behalf of the Board of Deans of Maastricht University, I congratulate you with the honor you have acquired. Of course, I would also like to congratulate your family, friends, and colleagues, and your supervising team, Professor Lerusso, Professor Maase, and Dr. Raffa. I would also like to thank the members of the degree committee for their presence and their contribution to this defense. Your contribution is always very important, and Maastricht University is very grateful for the work you're doing in this respect. We now come to the end of this session, and I declare this ceremony to be ended.